So you can see on the right is glycolysis, and if we start from glycogen, which is stored in the muscle, the first process is to break glycogen down into glucose 1-phosphate, and then from glucose 1-phosphate to glucose 6-phosphate. If we look to the left, we've got glucose, and glucose is in the blood. We've got in the diagram glucose traveling through the plasma membrane with the help of what's called a glucose transporter, the GLUT4. Glucose enters inside the cell, so into the cytosol of the cell, and from there an enzyme called hexokinase converts glucose to glucose 6-phosphate. So glucose 6-phosphate is really the beginning of glycolysis. So if you start from a glucose, it's converted to glucose 6-phosphate through hexokinase, and whereas glycogen has to be converted into glucose 1-phosphate and glucose 6-phosphate. There are a number of enzyme controlled reactions in glycolysis, and the end product is pyruvate or lactate. Researchers believe that lactate is the preferred product of glycolysis, as for every 1 pyruvate produced from glycolysis, there are 10 lactate produced. In the diagram we can see the 10 steps of glycolysis. We start off with a glucose molecule in this example, and we end with a pyruvate at the end. We'll go through some of the more important steps in glycolysis, and the key thing is that we don't need to remember each of the processes or reactions, and we don't need to remember the enzyme names. I'll go through some of the key steps, and I'll point out the key aspects that you may need to know for your in-class test. Step 1 of glycolysis. In this first process, we're going to take a glucose molecule and we're going to find how it's converted into glucose 6-phosphate. Also need to note that this process requires an ATP to be broken down, so it requires energy to be invested. So we can see that glucose is combined with an ATP on the enzyme hexokinase. We can see the difference between the glucose molecule and the resultant molecule is glucose 6-phosphate. That is a glucose molecule with the addition of a phosphate. Now this reaction is very, very important, because by putting a phosphate onto a glucose, in effect it traps it inside the cell. If you remember, glucose is transported inside the cell via the GLUT4 transporter. By adding the phosphate to the glucose to form glucose 6-phosphate, the glucose 6-phosphate is not recognizable to the GLUT4 transporters, so therefore it can't be transported back outside the cell. So this is an important process, and it's the one in which really starts glycolysis. So this requires an ATP, as we said, because we attach the phosphate, we take the phosphate off the end, we take the end phosphate away from ATP, and we combine it with glucose, and we get ADP as a result. So we get glucose 6-phosphate and ADP. This is the ATP investment phase. Let's look at step 3. This is where fructose 6-phosphate is converted into fructose 1,6-biphosphate. And again, this requires ATP to be invested. So as we can see, that fructose 6-phosphate is a molecule that contains 1-phosphate. So we combine our fructose 6-phosphate with an ATP on the enzyme phosphofructose fructokinase, or PFK for short. And the resultant reaction gives us fructose 1,6 biphosphate. This is important, as we can see that this molecule is a symmetrical molecule. That is, it has a phosphate on both the right hand side and a phosphate on its left hand side. So to recap, this process has cost us another ATP. So at this stage we are minus 2 ATP. Step 6 of glycolysis. We can see that the fructose 1,6 biphosphate has split into G3P and DHAP. That DHAP is then converted to G3P. So in step 5 we have glyceraldehyde 3-phosphate on the left and glyceraldehyde 3-phosphate on the right. So glyceraldehyde 3-phosphate, or G3P for short. So this is important because the fructose 1,6 biphosphate has been split into two, and each of these glyceraldehyde 3-phosphates contain a phosphate. So we'll see this in the next stage. So step 6 of glycolysis. So as we can see, we have a glyceraldehyde 3-phosphate. So remember, this is one of the two glyceraldehyde 3-phosphates that has been produced. We can see that this glyceraldehyde 3-phosphate has a phosphate. So it is brought together with NAD plus on the enzyme triose phosphate dehydrogenase. We don't need to know the name of this enzyme. It's just important that this process attaches another phosphate to each of the glyceraldehyde 3-phosphates. So what we have is a molecule on the left that has 2 more phosphates, and on the right that has 2 more phosphates. So we have a 1,3 biphosphoglycerate. The other thing is to note is that NAD plus has taken 2 hydrogens away from our glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate has lost 2 hydrogens in this reaction, but has gained a phosphate. Now let's look at this NAD plus and NADH reaction a little bit more closely. This reaction is what we call a redox reaction. That is a reduction oxidation reaction. And this involves the transfer of electrons from one molecule to another. So glyceraldehyde 3-phosphate loses electrons, and it loses electrons through the loss of hydrogens. So NAD plus takes 2 hydrogens away from glyceraldehyde 3-phosphate. Now, if we remember in class, we talked about how to remember between oxidation and reduction. We had the saying oil rig. Oxidation is losing, reduction is gaining, oil rig. So oxidation is when you've lost electrons. So glyceraldehyde 3-phosphate has been oxidized. NAD plus has gained electrons, so this has been reduced to NADH. So NADH is the reduced version of NAD plus. So just so that we don't have any confusion between electrons and hydrogens, on the right hand side we can see what a hydrogen atom looks like. It has a plus charge in the middle, and this is due to the proton, and it has a negative charge on the outside, which is due to its electron. So it has 1 proton and 1 electron. A hydrogen ion carries a positive charge because it still has its proton, but it's lost an electron. Because hydrogen is a very simple atom, it very easily loses its electrons, or it's very easy to take electrons away from it. So out of the glyceraldehyde 3-phosphates, the NAD plus takes the hydrogens away to get its electrons. Now let's look at this a little bit more detail. Here we have our NAD plus molecule, our nicotinamide adenine dinucleotide. The first thing that we should notice is encircled in the red is that we have a nitrogen atom, and this nitrogen atom carries a positive charge. It carries a positive charge because it's an electron short. That is, it has more protons than it has electrons. It's missing electrons, or missing one electron. So let's look at our reduced version of our NAD+, our NADH. So we can see here that it has an additional hydrogen, so it's gained the hydrogen, so it's taken a hydrogen from glyceraldehyde 3-phosphate, and that hydrogen is complete. It has one proton and one electron. The nitrogen no longer carries a positive charge, so that nitrogen has gained an electron. So we know that it's taken two hydrogens away from glyceraldehyde 3-phosphate. One of the hydrogens is combined at the top. An electron has been attached to nitrogen, and that must have come from the other hydrogen, so we must be left over with one hydrogen ion. That is a hydrogen that has lost its electron. It's lost its electron to nitrogen. So we have a hydrogen ion left from this reaction. So glyceraldehyde 3-phosphate has been oxidized, and NAD+ has been reduced to NADH, and a hydrogen ion is left over also. So key thing to remember and keep in mind is that NADH has gained 2 electrons. It has 2 more electrons than NAD+. This is important because this is our electron transporter. NADH is our electron transporter. So let's look at our next step that we're going to concentrate on. This step is step 7. So we have our 2 molecules of 1,3-biphosphoglycerate, and this is an important stage because we get the first of our ATP being generated. So remember earlier on, to get this far, it's already cost us 2 ATP. So let's have a look at this in a little bit more detail. We have our 1,3-biphosphoglycerate molecule with 2 phosphates attached to it. So we bring together ADP and our 1,3-biphosphoglycerate onto our enzyme, our phosphoglycerokinase. This process involves taking one of the phosphates off 1,3-biphosphoglycerate and combining it with an ADP, and this gives us an ATP. This occurs for each of the 1,3-biphosphoglycerate molecules, so we get 2 ATP generated in this process. What's the product of this reaction is our 3-phosphoglycerate, so this still has 1 phosphate left on it. Let's have a look in the next stage what happens to this phosphate. In step 10, we've got the next generation of ATP. So we had from step 7, we've got 2 ATP being produced. We've got the molecule is changed slightly in step 8 and 9, and then we have step 10 where we get some ATP back and we get the production of pyruvate. So we have our PEP. Our PEP molecule still has a phosphate attached to it. So we combine it with ADP and the enzyme pyruvate kinase, and that gives us an ATP. So we take the phosphate off our PEP, attach it to our ADP, and we get ATP back. Because we've got two PEP molecules, we get two ATP. And we get two pyruvates as a result. So we've started from our glucose molecule and we've ended at our pyruvate molecule, or our two pyruvate molecules, I should say. So in summary, we have, when we use glucose, we break it down into pyruvate, and this is called glycolysis. This process generates what's called NADH, and NADH is an electron carrier. And if we remember earlier on, each NADH molecule carries 2 electrons. We had 2 NADH being produced, so therefore we have 4 electrons being transported on 2 NADH molecules. We also got the generation of ATP. But how many ATP did we get? We know at the start, in the first few processes, it cost us ATP, so we were minus 2 ATP. Then our molecules split in 2, and we had the generation of 2 ATP in stage 7, and then another 2 ATP in stage 10 to give us 4 ATP being generated in total. Minus the 2 that we invested earlier on, that gives us a net gain of 2 ATP for this process so far. And it's important to note that this process occurs in the cytosol of the cell.